Hello, this is Jian Feng Ma from Institute of Plant Science and Resources, Okayama University in Japan. It is my great honor to receive the IFA Norman Borlaug Plant Nutrition Award this year. First of all, I would like to thank all my staff members, postdocs, students, and all collaborators for their hard work and strong support. I have been working in plant nutrition field for almost 40 years. As you know, plant nutrition is characterized by mineral nutrition. During last 3 decades, we focus on identification of transporters for different mineral elements, including essential, beneficial, and toxic elements. So far, we have identified a number of transporters involved in the uptake, root-to-shoot translocation, distribution, and distribution, mainly in rice and sometimes also in barley. For example, we identified 2 transporters required for silicon uptake in rice. These transporters are very important for silicon accumulation, high silicon accumulation in rice. They're polarly localized at the root exodermis and endodermis and form an efficient cooperative system for silicon uptake. We also found that in addition to silicon, similar system is also present for boron and manganese in rice. In addition, we also identified 3 transporters for preferential distribution of silicon to the panicles. They are localized at the different series in the node and are responsible for the intravascular transfer of silicon in rice. In response to environmental change, plants have developed a sophisticated system to cope with these changes through regulating the transporters at the transcriptional and protein levels. For example, we found OSNLAMP3, a manganese transporter, function as a switch. When manganese concentration in the soil is low, this protein is well expressed and is involved in the preferential distribution of manganese to the developing tissues such as new leaf and panicles. However, when manganese concentration in soil is high, this protein is immediately degraded to avoid the manganese toxicity. So based on the transporters we discovered, we sometimes also manipulate these transporters for biofortification. Rice is a staple food for half world's population, but it is a poor source of zinc and iron because rice has a low density of zinc and iron in the grain. It also contains high phytic acid in the grain. Recently, we found if we knock out some transporters localized at the tonoblast in the node, will result in increased concentration of zinc and iron in rice grain. We also found that knockout of a novel phosphorus transporter, SPDT, decreased concentration of phytic acid in rice grain. As I mentioned, rice is a staple food for us, but it is also a major source of our intake of toxic elements such as cadmium and arsenic. So far, we also have identified a number of transporters involved in the accumulation of these toxic elements in rice. Especially recently, cloned a gene which is involved in low cadmium accumulation in rice. We successfully bred a rice cultivar by introducing this gene to an elite rice cultivar by multiple backcrossing. So this provides a sustainable way to reduce cadmium accumulation in rice grain for our health. So transporter links mineral elements among soil, plants, and human health. I hope that our work will further contribute to better and safe crop production in future. Thank you for listening.